Hello friends, welcome back to my YouTube channel Journey with Vijay Kumar Srivastava. Today we will study about the topic genetic principles in hybrid seed production. So let's proceed. This slide refers about the hybrid seed, how it is produced and what is the importance of seed production. Hybrid seed is the F1 generation seed produced from a cross between genetically dissimilar parents. And the hybrid vigor or heterosis is the absolute phenomenon where expression of the characteristics such as growth related traits, flowering and yield fall outside the range of its parents with superiority. So whatever parents we are using, the hybrids will behave with high potential, having high yielding characteristics and vigor and other traits. Hybrid is produced by crossing between two or more genetically dissimilar parents. Pollen from male parents will pollinate, fertilize and set seeds in female parent. The male parent is known as pollen parent and female is known as seed parent. And the produce will be F1, which is known as hybrid seed. For production of a hybrid, crossing between two parents is important and the crossing process will result in heterosis. In self-pollinated crops, it is difficult to cross, but in cross-pollinated crops it is easier. Cross-pollination results in genetic heterogeneity and so wider adaptations. As cross-pollinated crops are concerned, we can refer this maize, pearl millet, sorghum, castor, etc., where the self-pollinated, where the hybrid seed production is done, like hybrid rice. As the picture has been shown, the male parent will pollinate the female parent and it will result in F1, which is known as hybrid seed. Now to study importance of seed production. Seed production refers about the multiplication of superior varieties or hybrids and its availability to masses. And it is important for maintenance of genetic purity, supplying high quality seeds, seeds having good germination status and having high viability, and it can maintain the excellent health status of seed and securing high yields and income levels to the farmers. The components of genetic principles in hybrid seed productions are use of parental lines, male sterility, self-incompatibility, role of stigma receptivity, pollen viability, emasculation and pollination process, use of genoecious sex forms, use of chemical and growth regulators, sex modifications through hormones and chemicals, and manipulation of environment for sex modifications. So we will study about details of each component. In hybrid seed production, three-line concept is used where three parental lines are used: A line, B line and R line. A line is female line, which is also known as male sterile line. Second one is B line, known as maintainer line. And third one is R line male, which is commonly known as restorer line. We will study in details about all lines and their importance in hybrid seed production. Hybrid seed production involves two steps. First one is maintenance of parental lines. As earlier discussed, we use three lines: A line, B line and R line. So its multiplication to be done in a required quantity. Second one is commercial hybrid seed production, where two lines, A line and R line, are used. Hybrid seed production requires a female plant in which viable male gametes are absent. So these are known as male sterile lines. And it may be naturally or it is created by removal of the male organs from the flowers. Hand emasculation is also done to make a plant devoid of pollen so that it can be used as a female parent, and this process is used in cotton hybrid seed production and okra seed production also, and some other crops also in vegetables where the artificial emasculation process is done. Another simple way to use a female line for hybrid seed production is to identify or create a line that is incapable to provide viable pollen. So male sterility is developed and these lines are used as a male sterile line. In some crops like maize, pearl millet, sorghum, rice, mustard, sunflower, castor, sorghum Sudan grass, etc., the female parent is a sterile plant, while male sterility is naturally or artificially developed. And some crops like okra, chili, cotton, emasculation process is followed. In hybrid seed production, the lines which are used for production are A line, which desirable characteristics are very important, as well as restorer R line, about which we will discuss in details in coming slides. Here we have shown the diagram how the hybrid seed production is done and how the lines are multiplied. If we are using A line and B line, it will result in multiplication of A line, which is male sterile line, and again this male sterile line A parent is multiplied with R line restorer line, then it will generate the hybrid seeds. A line is used as a male sterile line, B line is a maintainer line, and R line is restorer line, and each line definitions, requirement, characteristics will be discussed in detail. Here we will go through utilization of A line, B line, R lines in hybrid seed production. As we earlier discussed, A line and B line are used for the multiplication of A line, which results in male sterile line, and this A line, when multiplied with R line restorer line, it will result in the production of hybrid seed, which is male fertile and known as F1 hybrid seed. So this totally flow chart has given the details how the process incorporated in utilization of these lines and production of hybrid seed. Now we will discuss in detail about A line. A line is male sterile line, and this sterility is of different kinds like cytoplasmic male sterile line, genetic male sterile line, and cytoplasmic genetic male sterile line. This A line is maintained by crossing with B line in an isolated plot, and the result of crossing between A line and B line will be production of A line, multiplication of A line, which will be male sterile. A line and B lines are known as isogenic lines, and the difference is of only male fertility in B line, while the A line is male sterile line. And the A line should have desirable characteristics which are very important to be incorporated in hybrid seed production. These A lines should have absence of pollen grains or rudimentary pollens, and it should have characteristics of abortive anthers not to produce any functional pollen grains, and it should be highly synchronized and having good matching with the R line, and this should be stable and complete male sterility and no effect of environmental while production, and the line should have good out crossing ability with R line and good combining ability, and the desirable agronomic characteristics are as concerned it should have well developed floral test of outcrossing, the stigma receptivity, and as earlier discussed about combining and outcrossing is having of best level, it will result in good crossing and seed setting and production of this F1 hybrid seed. And A line should have wide generation spectrum and a strongly restored characteristics. B line is known as maintainer line, which is used to maintain the sterility in A line, multiplication of A line, and this is used as a pollen parent. The B line is maintained just like normal varieties by following the production conditions of isolation and quality field standards. Just by selfing, it can be produced multiplied, while when the B line is crossed with A line, it will result in A line production, which will be the male sterile, and the B line is isogenic line of A line, where the difference persist in male sterility and fertility. The B line is male fertile while A line is male sterile. The B line having the characteristics of well developed floral traits with large pollen load, because the pollen is needed to multiply the A line in a sufficient quantity. It will result in good seed setting and fertilization. And the B line should have normal anthers, functional pollen grains, and good seed setting on selfing, and it should have good combining ability with A line. And A and B lines, the nucleus should be same, and only cytoplasm will vary with regards to sterility and fertility. R line is known as restorer line and also called as fertility restorer or pollinator line. R line is having the characteristics of fertility restoration gene, and this line has restorer gene in nucleus to restore fertility of A line. R line is having the entirely different characteristics to A and B line, and this is used to cross male sterile lines to produce hybrid seeds. R line has very high specific combining ability effects, and it is used to produce hybrid seeds, so it plays very important role for the productivity and restoring the fertility. Restorers plays an important role both in increasing heterosis as well as in increasing hybrid seed yield in seed production plots. The R line is maintained just like normal varieties by selfing, growing in isolated conditions with field standards where the quality checks, rogging and seed standards are compulsorily followed. And the multiplication of F1 hybrid by the crossing of A line with R line results in hybrid seed production. In hybrid seed production, A and R lines are used. R line is very important as the yields and productivity is concerned. The desirable characteristics of R line may be defined as having normal anthers with high functional pollens. There should be a strong and stable restoration capacity over seasons and locations. The R line should have highly vigorous plant growth habit with large number of spikelets to produce large number of anthers and pollen load supply. And there should be complete dehiscence or dispersal of anthers to the A line for better crossing ability. And there should be high amount of residual pollen and abundant pollen producing capacity. As the pollen grains supplied to the female sterile line A is concerned, it should have profuse tillering, synchronous heading and concentrated blooming of spikelets to supply the pollen grains for the longer time. And the R line should have multiple disease and pest resistance to incorporate in hybrid seed, and R line should have good outcrossing and combining ability, and there should be acceptable grain quality and high outcrossing rate. To get the clear understanding of A line, B line and R line, we have shown here the pictures of hybrid bajra or pulse millet seed production fields, and we can get the differences how the R line, A line are shown. As earlier we had discussed that A line and B line are isogenic lines and having the difference of only male fertility. In fields, the B line if found in A line is known as pollen shedder, and we can see how the difference between A line and B line. The B line is having the anther boxes with supply of pollen, while A is having the stigma, and there will be no functional pollen grains or anther boxes in A line. Here also some pictures of R line and A line in sunflower fields and maize fields. R line in sunflower is having the functional pollen grains and different pollen loaded anthers, while in case of A line it will be sterile. In case of maize, the tassels are known as male parent, which are having the anther boxes and supply of pollen grains, while the silk is known as female parent. In case of maize, the detasseling process is done to make the female sterile, where the tassels will be removed and the pollen grains falling from these tassels are pollinating the silk. In case of SSG also, we have shown the parents of male and R line. This picture represents the use of A and R line in hybrid seed production. Both A and R lines are planted in a row ratio sowing, where the pollinator is supplying the pollen to pollinate female line, which is male sterile line, and the A line is the seed parent, which is harvested, and the F1 seed is used as a hybrid. Another genetic component of hybrid seed production is male sterility. Male sterility is defined as an absence of non-functional pollen grains in plant or characteristics of incapability of plants to produce or release functional pollen grains. The male sterility prevents self-pollination, it facilitates cross-pollination, promotes heterozygosity, and it is used or exploited in agriculture crop plants for hybrid seed production. The male sterility is of different types, like genetic male sterility, cytoplasmic male sterility, and cytoplasmic genetic male sterility. In deep details, the genetic male sterility is controlled by mutations in nuclear genes, and genetic male sterility is controlled either by dominant or recessive genes. The male sterile gene is maintained by crossing male sterile line with a heterozygous male fertile line, means B line, and the GMS line, where the A is maintained by back crossing with the heterozygous B line, which is known as maintainer line. A line is the seed parent, and in case of genetic male sterility, there will be 50% fertile and 50% male sterile plants. So, when we are using the genetic male sterile lines in hybrid seed production, we have to rogue out 50% of male fertile plants. The crops like cotton, the GMS lines are used for hybrid seed production, where the roguing is very important to rogue out the male fertile plants. Second is cytoplasmic male sterility, which is governed by extranuclear genes which are present in mitochondrial genome, and these show non-Mendelian inheritance patterns and are under the regulation of cytoplasmic factors. Third type of sterility, cytoplasmic genetic male sterility, CGMS lines, which is controlled by both the nuclear and cytoplasmic genes. And here the male sterile line is maintained by crossing with a maintainer line that has the same genome as that of male sterile line, but it contains or carries normal fertile cytoplasms. And the fertility restoration is done by the fertility restorer nuclear gene, RF genes. Another genetic component of hybrid seed production is self-incompatibility, which is a mechanism that prevents self-fertilization through recognition of self-pollen on a stigma on the same flower in the same plant or that of other plants of the same genotype. But in this case, the pollen from other plants which are carried by wind, insects and other vectors deposit on a stigma of such flowers and sets seeds by the pollination. Therefore, we can say the self-incompatibility prevents self-fertilization and facilitates and promotes cross-fertilization. And the self-incompatibility is observed in both hermaphrodite and homomorphic flowers. And the self-incompatibility response is generally controlled by one or more multi-allelic loci and relies on the series of complex cellular interactions between self-incompatible pollen and pistils. Now to study stigma receptivity and pollen viability. Stigma is the part of flower or organ of the flower on which pollen lands to facilitate fertilization and consequently seed formation. There are variations in stigma morphology which supports pollen germination and tube growth in the compatible pollen. And the longer stigma receptivity in cytoplasmic male sterility is a desirable trait that favors higher seed yield. Means if the stigma is having longer receptivity, it will result in good seed setting and ultimately good yielding. And it is important both for manual or natural pollination by vectors like wind, insects, etc. And generally, stigma becomes receptive at the end of flower opening, which is known as anthesis. And the duration of stigma receptivity is usually longer in cytoplasmic male sterile lines compared to their fertile counterparts. Now the pollen viability, where the pollen maturation to its release and dispersal, germination on a receptive stigma, and fertilizations are essential steps to plan the timing for hybridization for successful hybrid seed production. The response of pollen during its mobile phase, like after its release and detachment from the another flower of the anther of the pollen parent, is also important as pollen passes through different environmental conditions. The duration of pollen viability varies greatly among crop species and varies from few minutes to hours. Pollen viability or longevity depends on several climatic factors like temperature, humidity, etc., as well as pollen vigor that might be influenced by the maturation and disease status of the plant. The knowledge of both stigma receptivity and duration of pollen viability is important for successful hybrid seed production. Now to go through emasculation, pollination and detasseling. What is emasculation? Removal of male organs or stamens or anthers are called emasculation. In bisexual flowers, emasculation is essential to prevent cell pollination, and the stamens are manually removed from a bisexual flower in a female plant before flower opening or anthesis. And this system is feasible when the male and female parts of a single flower or plants are separate, and this is practiced in bisexual perfect flowers where the androecium could be removed easily. Now the pollination, where the pollen of desired male line is dusted manually on the stigma of the emasculated flowers in female parents to facilitate pollination. And this technique is commercially feasible in crops that have large conspicuous flower. Easy removal of stamens and anthers is possible, and there is high seed set rate for pollination, low seed rate per hectare and high cost of hybrid seed. And the crops like cotton, okra, tomato, brinjal, chili, melon, etc. crops are having the emasculation and pollination process in hybrid seed production. And this technique requires trained labor for emasculation, pinching, bagging, pollen collection and pollination, adding to the cost of hybrid seed. And the male and female rows are grown in recommended row ratio or block methods, where the fruit set on female line is harvested for hybrid seed extraction. So, in this case, it is vital to know the floral biology, flowering time, crop morphology, synchronization process in flowering in the parental lines for emasculation and pollination of the seed parents. In hybrid seed production of some crops like maize, detasseling is done to develop sterility, and wind pollination occurs naturally. And this detasseling is defined at the process of removal of female tassels before anther bursting to make female pure sterile line, and it must be done in 7 to 10 days between when the tassel is ready to emerge and pollinate. Here are the some pictures which represents the emasculation and pollination process in cotton and detasseling activity in maize crop. Other genetic components are gynoecious sex forms in hybrid seed production, which are commercially exploited in hybrid seed production activities of cucurbits like cucumber, sponge gourd, bitter gourd, muskmelon, etc., where the female and male rows are planted in a specific row ratio under favorable climatic conditions. The female parents are pistillate flowers, and pollination is done by insects like honeybee and wasps, where the good population is maintained of the honeybee to ensure good fruit, seed set, and seed recovery. The male parent line is maintained by selfing, and the female lines, gynoecious lines, are maintained by inducing the staminate flowers with the application of silver nitrate at the rate of 200 rppm concentration. The homozygous lines are produced in strict isolations, and the gynoecious lines are crossed with the monoecious male parent to produce F1 hybrid. And the gynoecious lines are unstable under high temperature and long photoperiod conditions. Hence, gynoecious cucumber didn't become popular in tropical countries. Other components are use of chemicals and growth regulators in hybrid seed production. The hybrid seed can be produced by inducing femaleness and maleness with the application of chemicals and growth regulators. There are some chemicals which are inducing femaleness, like use of auxins, and there are some chemicals which induce maleness, like silver nitrate, abscisic acid, gibberellic acid, etc. In case of cucumber, these silver nitrates are found to be potent inhibitor of ethylene, and the gibberellic acid spray leads to excessive elongation and weakening of plant, and there may be increased number of malformed male flowers with less population. So these chemicals are used in extensive ways to induce femaleness and maleness to get the desired yields. As the genetics is concerned, sex modifications is also done through hormones and chemicals, and it can be modified to a considerable extent by environmental and introduced factors such as mineral nutrition, photoperiod, temperature, and phytohormones. And these phytohormones are the most effective agents for sex modifications. The morphological differences in various sex types and their specific metabolic characteristics results from the possession of specific patterns of proteins, enzymes, and other molecules. And the modification of sex expression in cucurbits has been induced both by changing the environmental conditions as well as applying treatments with growth regulators. Auxin treatment increases femaleness, while gibberellins cause a shift towards maleness, and the application of plant growth regulators is reported to alter sex expression and flower sequence in cucurbits. As a genetics is concerned, the manipulation of environment for sex modification is applied in seed production, and the environment has a strong influence on sex modification. In case of rice, the expression of male sterility and its restoration is influenced by environment sensitive genetic male sterility (EGMS), and it has been further classified into photosensitive and thermosensitive genetic male sterility genes. The hybrids developed using these systems are called two-line hybrids, as no maintainer is required for the multiplication of the female line, and it is controlled by the environment. Castor is a monoecious plant where the staminate flowers and pistillate flowers are located at different positions in a raceme, and there are genotypes with a predominantly higher proportion of pistillate flowers, which are governed by both genetic and environmental conditions. Low temperature conditions promotes pistillate plants in particular genotypes that reverse to monoism with increasing temperature and higher order of branches. Maximum female sex expression is seen when the daily temperature during raceme formation and development is less than 30 degrees centigrade. Female lines are multiplied at higher temperature conditions that induce temperature sensitive interspersed staminate flowers, and hybrid seed production is taken up at relatively low temperature condition in which this interspersed staminate flowers is not formed in a pistillate line. Therefore, two-line hybrid system production is also possible in castor. Male sex expression of several plant species is favored at high temperatures and female sex expression at low temperatures. In case of tomato, male sterile mutants develop male sterile flowers at a temperature of 30 degrees centigrade and normal flowers at lower temperature. So here we can see the effect of environment on sex modification, and it is used accordingly in seed production activities. This is the end of my presentation of the topic genetic principles in hybrid seed production. Thanks, and see you soon again shortly with new presentations.